Welcome back to Concepts of Biology. We'll continue to read Chapter 2, Section 2.3, Biological Molecules. Nucleic acids. Nucleic acids are key macromolecules in the continuity of life. They carry the genetic blueprint of a cell and carry instructions for the functioning of the cell. The two main types of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA is genetic material found in all living organisms, ranging from single-celled bacteria to multicellular mammals. The other type of nucleic acid, RNA, is mostly involved in protein synthesis. The DNA molecules never leave the nucleus, but instead use an RNA intermediary to communicate with the rest of the cell. Other types of RNA are also involved in protein synthesis and its regulation. DNA and RNA are made up of monomers known as nucleotides. The nucleotides combine with each other to form a polynucleotide, DNA or RNA. Each nucleotide is made up of three components: a nitrogenous base, a pentose or 5-carbon sugar, and a phosphate group. Each nitrogenous base in a nucleotide is attached to a sugar molecule, which is attached to a phosphate group. Figure 2.22. A nucleotide is made up of three components: a nitrogenous base, a pentose sugar, and a phosphate group. DNA double-helical structure. DNA has a double-helical structure. It is composed of two strands or polymers of nucleotides. The strands are formed with bonds between phosphate and sugar groups of adjacent nucleotides. The strands are bonded to each other at their bases with hydrogen bonds, and the strands coil about each other along their length, hence the double helix description, which means a double spiral. Figure 2.23. The double helix model shows DNA as two parallel strands of intertwining molecules. The alternating sugar and phosphate groups lie on the outside of each strand, forming the backbone of the DNA. The nitrogenous bases are stacked in the interior like the steps of a staircase, and these bases pair. The pairs are bound to each other by hydrogen bonds. The bases pair in such a way that the distance between the backbones of the two strands is the same all along the molecule. Let us review our key terms and summarize Chapter 2. Acid, a substance that donates hydrogen ions and therefore lowers pH. Adhesion, the attraction between water molecules and molecules of a different substance. Amino acid, a monomer of a protein. Anion, a negative ion formed by gaining electrons. Atomic number, the number of protons in an atom. Base, a substance that absorbs hydrogen ions and therefore raises pH. Buffer, a solution that resists a change in pH by absorbing or releasing hydrogen or hydroxide ions. Carbohydrate, a biological macromolecule in which the ratio of carbon to hydrogen to oxygen is 1 to 2 to 1. Carbohydrates serve as energy sources and structural support in cells. Cation, a positive ion formed by losing electrons. Cellulose, a polysaccharide that makes up the cell walls of plants and provides structural support to the cell. Chemical bond, an interaction between two or more of the same or different elements that results in the formation of molecules. Chitin, a type of carbohydrate that forms the outer skeleton of arthropods such as insects and crustaceans and the cell walls of fungi. Cohesion, the intermolecular forces between water molecules caused by the polar nature of water, creates surface tension. Covalent bond, a type of strong bond between two or more of the same or different elements, forms when electrons are shared between elements. Denaturation, the loss of shape in a protein as a result of changes in temperature, pH, or exposure to chemicals. Deoxyribonucleic acid, DNA, a double-stranded polymer of nucleotides that carries the heredity information of a cell. Disaccharide, two sugar monomers that are linked together by a peptide bond. Electron, a negatively charged particle that resides outside of the nucleus in the electron orbital, lacks functional mass and has a charge of -1. Electron transfer, the movement of electrons from one element to another. Element, one of 118 unique substances that cannot be broken down into smaller substances and retain the characteristics of that substance. Each element has a specific number of protons and unique properties. Enzymes, a catalyst in a biochemical reaction that is usually a complex or conjugated protein. Evaporation, the release of water molecules from liquid water to form water vapor. Fat, a lipid molecule composed of 3 fatty acids and a glycerol triglyceride that typically exists in a solid form at room temperature. Glycogen, a storage carbohydrate in animals. Hormone, a chemical signaling molecule, usually a protein or steroid, secreted by an endocrine gland or group of endocrine cells. Acts to control or regulate specific physiological processes. Hydrogen bond, a weak bond between partially positively charged hydrogen atoms and partially negatively charged elements or molecules. Hydrophilic, describes a substance that dissolves in water, water-loving. Hydrophobic, describes a substance that does not dissolve in water, water-faring. Ion, an atom or compound that does not contain equal numbers of protons and electrons and therefore has a net charge. Ionic bond, a chemical bond that forms between ions of opposite charges. Isotope, one or more forms of an element that have different numbers of neutrons. Lipids, a class of macromolecules that are nonpolar and insoluble in water. Litmus paper, filter paper that has been treated with a natural water-soluble dye so it can be used as a pH indicator. Macromolecule, a large molecule often formed by polymerization of smaller monomers. Mass number, the number of protons plus neutrons in an atom. Matter, anything that has mass and occupies space. Monosaccharide, a single unit or monomer of carbohydrates. Neutron, a particle with no charge that resides on the nucleus of an atom, has a mass of 1. Nonpolar covalent bond, a type of covalent bond that forms between atoms when electrons are shared equally between atoms, resulting in no regions with partial charges as in polar covalent bonds. Nucleic acid, a biological macromolecule that carries the genetic information of a cell and carries instructions for the functioning of the cell. Nucleotide, a monomer of nucleic acids, contains a pentose sugar, a phosphate group, and a nitrogenous base. Nucleus, in chemistry, the dense center of an atom made up of protons and, except in the case of a hydrogen atom, neutrons. Octet rule states that the outermost shell of an element with a low atomic number can hold 8 electrons. Oil, an unsaturated fat that is a liquid at room temperature. Periodic table of elements, an organizational chart of elements indicating the atomic number and mass number of each element, also provides key information about the properties of elements. pH scale, a scale ranging from 0 to 14 that measures the approximate concentration of hydrogen ions of a substance. Phospholipid, a major constituent in the membranes of cells, composed of 2 fatty acids and a phosphate group attached to the glycerol backbone. Polar covalent bond, a type of covalent bond in which electrons are pulled toward one atom and away from another, resulting in slightly positive and slightly negative charged regions of the molecule. Polypeptide, a long chain of amino acids linked by peptide bonds. Polysaccharide, a long chain of monosaccharides, may be branched or unbranched. Protein, a biological macromolecule composed of one or more chains of amino acids. Proton, a positively charged particle that resides in the nucleus of an atom, has a mass of 1 and a charge of +1. Radioactive isotope, an isotope that spontaneously emits particles or energy to form a more stable element. Ribonucleic acid, RNA, a single-stranded polymer of nucleotides that is involved in protein synthesis. Saturated fatty acid, a long-chain hydrocarbon with single covalent bonds in the carbon chain. The number of hydrogen atoms attached to the carbon skeleton is maximized. Solvent, a substance capable of dissolving another substance. Starch, a storage carbohydrate in plants. Steroid, a type of lipid composed of 4 fused hydrocarbon rings. Surface tension, the cohesive force at the surface of a body of liquid that prevents the molecules from separating. Temperature, a measure of molecular motion. Trans fat, a form of unsaturated fat with the hydrogen atoms neighboring the double bond across from each other rather than on the same side of the double bond. Triglyceride, a fat molecule, consists of 3 fatty acids linked to a glycerol molecule. Unsaturated fatty acid, a long-chain hydrocarbon that has 1 or more than 1 double bonds in the hydrocarbon chain. Van der Waals interaction, a weak attraction or interaction between molecules caused by slightly positively charged or slightly negatively charged atoms. Chapter Summary. In Section 2.1, the building blocks of molecules. Matter is anything that occupies space and has mass. It is made up of atoms of different elements. All of the 92 elements that occur naturally have unique qualities that allow them to combine in various ways to create compounds or molecules. Atoms, which consist of protons, neutrons, and electrons, are the smallest units of an element that retain all of the properties of that element. Electrons can be donated or shared between atoms to create bonds, including ionic, covalent, and hydrogen bonds, as well as van der Waals interactions. Section 2.2. Water. Water has many properties that are critical to maintaining life. It is polar, allowing for the formation of hydrogen bonds, which allows ions and other polar molecules to dissolve in water. Therefore, water is an excellent solvent. The hydrogen bonds between water molecules give water the ability to hold heat better than any other substance. As the temperature rises, the hydrogen bonds between water continually break and reform, allowing for the overall temperature to remain stable, although increased energy is added to the system. Water's cohesive forces allow for the property of surface tension. All of these unique properties of water are important in the chemistry of living organisms. The pH of a solution is a measure of the concentration of hydrogen ions in the solution. A solution with a high number of hydrogen ions is acidic and has a low pH value. A solution with a high number of hydroxide ions is basic and has a high pH value. The pH scale ranges from 0 to 14, with a pH of 7 being neutral. Buffers are solutions that moderate pH changes when an acid or base is added to the buffer system. Buffers are important in biological systems because of their ability to maintain constant pH conditions. In Section 2.3, Biological Molecules. Living things are carbon-based because carbon plays such a prominent role in the chemistry of living things. The 4 covalent bonding positions of the carbon atom can give rise to a wide diversity of compounds with many functions, accounting for the importance of carbon in living things. Carbohydrates are a group of macromolecules that are a vital energy source for the cell, provide structural support to many organisms, and can be found on the surface of the cell as receptors or for cell recognition. Carbohydrates are classified as monosaccharides, disaccharides, and polysaccharides depending on the number of monomers in the molecule. Lipids are a class of macromolecules that are nonpolar and hydrophobic in nature. Major types include fats and oils, waxes, phospholipids, and steroids. Fats and oils are a stored form of energy and can include triglycerides. Fats and oils are usually made up of fatty acids and glycerol. Proteins are a class of macromolecules that can perform a diverse range of functions for the cell. They help in metabolism by providing structural support and by acting as enzymes, carriers, or as hormones. The building blocks of proteins are amino acids. Proteins are organized at 4 levels: primary, secondary, tertiary, and quaternary. Protein shape and function are intricately linked. Any change in shape caused by changes in temperature, pH, or chemical exposure may lead to protein denaturation and a loss of function. Nucleic acids are molecules made up of repeating units of nucleotides that direct cellular activities such as cell division and protein synthesis. Each nucleotide is made up of a pentose sugar, a nitrogenous base, and a phosphate group. There are 2 types of nucleic acids, DNA and RNA. This is the end of Chapter 2. In our next section, we will go to Chapter 3, Cell Structure and Function.